we are always excited to tell the story of Sigilon. We say that we have the courage to revolutionize medicine and create a first-in-category therapeutics to treat patients with chronic disease. Imagine restoring the cell function. Imagine replacing the deficient proteins and producing those proteins in a programmable fashion. Imagine shielding cells from the immune rejection and avoiding fibrosis. Imagine really not interacting with the host genome, and on top of that, delivering off-the-shelf products. That's what we are doing, that's Sigilon is about. And we believe with that to overcome the limitations that exist today for chronic diseases. I consider myself a chronic disease expert. I've been in the industry for over 25 years, have been a clinician over 20 years in the clinic, and dealing with multiple different types of chronic diseases. And the minute-to-minute regulation, as well as the no peace of mind that patients with chronic disease have, they should get better treatments now that they are doing so much better. So a platform like we have, and we call shielded living therapeutics, harness the cell's really sophisticated machinery, is how we say that the cells then can do what the cells are designed to do. We start with a really broad pipeline. Again, chronic disease, we have to start, and having a product platform, we start with a broad one, and we are targeting our first IND CTA filing the end of this year, 2019, with followed by 2 or 3 others in the next 2 years, representing a big opportunity. We had signed last year a corporate deal with Lilly for finding a shielded living therapeutic products for type 1 diabetes. We received an upfront commitment of $75 million and more than $400 million downstream payments in addition to royalties. It's really a multi-billion-dollar opportunity that we can, by really revolutionizing the way of patients being treated. Let me introduce you to the shielded living therapeutics. The elements of our platform. First, we engineer the novel engineered human cells. We then encase those cells into what we call the proprietary immune biocompatible matrix. And let me explain you how this works. Imagine a balloon. You have a balloon, and inside of the balloon, you put a gel. So in that gel is where the cells will be residing, so that we don't want the cells to be flying around. We don't want the cells to spend too much energy and not produce the proteins that we want the cells to be producing. And the balloon gives the protection of the immune rejection and gives the protection of the fibrosis, while allows oxygen and nutrients to be in the capsule, really preserving the cell to be long living over there. We then place those cells into the body without the need of immune suppression, where the cells, again, will provide potential functional cure for many diseases, produce hormones, factors, enzymes, and antibodies. Just a small details, of course, we don't have too much time. On the cell platform, it's very important how to select the cell line, and all of these criteria that you're reading there are very important. So the cells, they will not overgrow. They will be really cells that we can utilize in multiple other proteins. As far as today, we have literally expressed more than 10 different proteins using one cell line. Of course, for diabetes is a different one. For all others, we are using the same cell line. We then go over many ways of engineer the cells, and you can see in the right graph how all of these optimization increase production. So in a very short time, less than a year, we can really go and increase the production by 100 times. If we look into what is this matrix about? And of course, you probably read, we have what we call a Afibromer technology. That is a technology that was developed by Bob Langer and Dan Anderson at MIT over a decade of work. And basically, it's a small molecule screening, and where they conjugate the biomaterials alginate with other polymers and other surfaces. And it's important, we then at Sigilon, we optimize this matrix, which is the inner compartment, in many ways, so that you can see the cell type, the components, the way the cell is being architectured, the density, to really focus on how can we get the best cell viability and the cell protein output. The Afibromer only goes into the outer space and the outer compartment of the matrix. And here I can show you the horizontal line shows all the experiments, 1, 2, and 3. On top of that line is without utilizing a Afibromer. And you can see an extensive scar tissue formation and really fibrosis. Down in those 3 different experiments, when we coat those surfaces or when we encapsulate those cells using spheres treated with a Afibromer, you can not see at all any fibrosis in any of the experiments. So those really were seminal, proving that we do have the cells being protected inside of that capsules. Where we put, in the last Cell and Med meeting in California, I didn't show that slide, and we are now have worked with a lot of surgeons. So it's a brief laparoscopic procedure under 30 minutes, where the capsules are infused or either intraperitoneally or into the lesser sac cavity. So it's a very simple, very quick procedures. Of course, we are working on other sites of implantation, including subcutaneous, including ocular, and the CNS as well. So what we have, all this data that we developed over the last 18 months, where we can see in the first one, the cells, again, we saw that they were protected, and here we are proving that they are efficacious. So the proteins, not only they leave the capsules, but also they get being absorbed throughout the body, and you see the bleeding time going down to normal levels and not in anything significantly different from the wild type. Also very important, in the middle graph, you have the controllability, which means, can we define the dose we want to have? In hemophilia, of course, sure, you have seen some of the gene therapy trials where you can find activity level of 20 at the same time, at the same cohort, you may have activity level going above 200% without exactly understanding what is going on. Here, we control the number of cells, the number of capsules, the protein output that we have in those cells and in those capsules, that we define exactly what level of activity we want to achieve in each of the patient's segments. And also one of the publications that we had showing over a year of controlling glucose metabolism in mice when we treat those capsules with a Afibromer, and we have the cells there doing their work. So all of these publications I mentioned before, Bob Langer, of course, probably you all know him, more than 1,600 patents, more than 40 companies being founded. When I was talking to him, he said, Rogerio, we never had 6 publications in Nature with one technology, so we are very proud. The last one is the one in the right, where you are seeing primates, and after 4 months that those capsules have been implanted in the lesser sac, you can see crystal and clear those capsules, those spheres, without any fibrosis. In addition to that, last year we were named as one of the top 10 emerging technologies of the year and we were really proud of this. So with this product platform, where can we go and where can we apply this? We could go in many directions, either systemic or local and regional. In the systemic, we could also have a continuous delivery of protein. Best example, hemophilia, where we continuous delivering protein, or through sense and respond. Best example, diabetes. Glucose rising levels trigger the cell to be producing insulin. But you have many other types of sense and respond, and we also work with lots of synthetic biology experiments and experts to really be developing a smart cell that will be inside of the capsule, so that will be long-living in the environment. So we have selected as initial targets three group of diseases that are very, very close to my heart. Hemophilia, I have been, I would say, very lucky to learn a lot in that community, particularly in my last job at Bioverative. The one thing that I can tell is that, and you heard this earlier today as well in one of the presentations, the many troughs and levels of factors are not sufficient for those patients. Patients that still have a very minimal number of bleeds throughout the year, 20 years later, they develop joint problems. So they need to have a different therapy, something that could be really continuous delivery. Lysosomal storage disorders, I was the first doctor in Brazil to be treating Gaucher disease and use enzyme replacement therapy, Serodase and Serozyme, back in 1991, a long time ago. And I learned with these patients that 60% of the patients with lysosomal storage disorders, they have a CNS component that, unfortunately, the enzyme replacement do not cross the blood-brain barrier, and we have to do something. We have to really do something. In addition to that, people don't remember, but when the enzyme replacement therapy schedules or dose were defined, the biweekly dose was defined because of was the only way to be able to treat patients with certain kind of adherence and you still get some good results, but not is the ideal one. So you'll see in a second why I'm mentioning this. And of course, type 1 diabetes, more than 1 million people in the United States with type 1, is still representing a lot of things to be done, no peace of mind. I can tell a little bit about this. I was in a clinic that I had more than 500 patients with type 1, and I have type 1 myself for only 45 years. I have a sensor, I have a pump, and I usually say that when I was diagnosed with type 1, means I lost the pancreas. Now replace the pancreas by my brain and think everything that I need to think in order to do really a good glucose control. Now I just want to replace the brain because the brain is really tired. I want to replace the brain by my cells. So those are the data that we produce in-house for each of these groups of disease. Of course, in 10 minutes, I could not show all the data. I present some of those in previous meetings, but you have really sustaining plasma, correction of the deficiencies. This in the middle is a very recent data that we did in MPS 1. We are extremely excited with this. Some of these results are equal or better than the initial results that I saw at Genzyme where I was for 20 years, when we were starting to work with diseases like MPS 1 and Fabry disease. And the last one in the right, after retrieving the capsules from the mice that have been implanted for 4 months, we then ex vivo tested if the cells were still functioning well to control glucose, and they were controlling, and they were still alive and functioning well. So this is what I said about the difference between the troughs and peak and really roller coaster to what exactly the Sigilon continues delivering. I think that one of the gene therapy companies showed exactly that, and yes, we have that similarity in terms of gene therapy goal. There are some differences, though, versus many of the new modalities. There is no pre-existing immunity for our patients. We don't have a capsid. The capsule does not provoke any immune reaction. We don't need any immunosuppression. Also, we have the ability to redose. And I also was very happy to hear in the previous presentation, someone saying that patients need to be informed that when they receive a shot for gene therapy, this is a one and done, but also excludes them to try something else or another gene therapy or another something. Here, it is not the case. If they really try to have shielded delivery therapeutics, they could have a redose, they could try something else. They are not, again, interacting with their own host genome. So I think in terms of safety, we have a very high bar that we are very happy, and we continue to work on this. Very quickly, mentioning the last meeting I was presenting at MESA, we are continuing progressing with our pipeline. Again, I mentioned the IND and CTA filing at the end of the year, and progressing also with the other rare blood disorders, hemophilia B, factor VII. And in the lysosomal storage disorders, we had really very exciting data, both on Fabry and MPS I. And of course, our collaboration with Lilly is really on track as we were expecting. One note, and one very quick note about the manufacturing, and this has been something that this group here is debating for a long time. Off the shelf, we need to think, how can we access patients globally? Patients with diseases, with chronic disease, with rare disease, they must have access. And I think that one solution like this could be applied throughout the world. I just want to finish by really thanking my great team, not only the ones that are appearing here in this slide, but the ones that are in Cambridge, and because they are really putting a lot of work. We have now more than 80 people working at Sigilon, and we are only focused to be fast so patients cannot wait. Thank you so much.